Stem cells were first discovered in the 1950s. Scientists discovered that bone marrow contains two different types of cells. One of the cells was found to be able to generate their own bone, cartilage, and fat cells. About 60 years later, and we have learned a lot from stem cells in all of their own different types. More recent, embryonic stem cells have been in the spotlight for some time due to their abilities and ethical concerns. Even more recent, we have made a discovery called induced pluripotent stem cells. Hello there, my name is John Gorman, and I'm going to guide you through what induced pluripotent stem cells are compared to embryonic stem cells, how the induced pluripotent stem cells are made or created, and the advantages and disadvantages of them. First, embryonic stem cells are taken from the inner mass cells of a young human embryo. The embryos themselves are made from the development of eggs that have been fertilized in an in vitro fertilization clinic and then donated for research, with consent of the donors, of course. Junying Yu and James A. Thompson has stated that the embryo at the time of extraction is only about 100 cells. The very young embryo contains two parts, an outer layer that forms a placenta and supports the embryo, and an inner layer of cells. Some of the inner cells are pluripotent, meaning they can make every type of the 220 different types of cells in the body, such as heart muscle cells, blood cells, or nerve cells. However, to generate the specific types of cells, scientists try to control the chemical composition and insert the specific genes necessary to create the type of cell that they need. Another ability of embryonic stem cells is that they are able to replicate themselves under specific conditions. This allows embryonic stem cells to be very dynamic and allows them to be useful tools for research and clinical use. On the other hand, induced pluripotent stem cells are taken from skin or blood cells that have been genetically reprogrammed to resemble an embryonic stem cell. This allows the induced pluripotent stem cells to act nearly identical to the embryonic stem cells by still being able to develop into all different types of stem cells needed for clinical purposes. I have a quote here from Charles Goldwhite. In 2006, researchers at Kyoto University in Japan identified conditions that would allow specialized adult cells to be genetically reprogrammed to assume a stem cell-like state. These adult cells, called induced pluripotent stem cells, were reprogrammed to an embryonic stem cell-like state by introducing genes important for maintaining the essential properties of embryonic stem cells. The large-scale production of embryonic stem cells has been limited due to the problem of obtaining embryonic tissues and the isolation of rare cell types. Scientists have then been exploring other ways to create pluripotent stem cells. The discovery in 2006 opened the doors that scientists have been looking for. The induced pluripotent stem cells are adult somatic cells that are genetically reprogrammed to emulate the embryonic stem cells. One method to do this is through nuclear reprogramming. I have another quote from Charles Goldwhite that states, "This approach involves taking mature somatic cells from an adult and introducing the genes that encode critical transcription factor proteins, which themselves regulate the function of other genes important for early steps in embryonic development. These factors were chosen because they were known to be involved in the maintenance of pluripotency. The newly created induced pluripotent stem cells were extremely similar to the embryonic stem cells and were established after a few weeks. Although this original strategy is inefficient in yielding induced pluripotent stem cells, before the induced pluripotent stem cells can be considered for use clinically, the efficiency of the making of them must improve. To do so, scientists used retroviral vectors. A retrovirus is an RNA virus that allows it to replicate in a host cell and then create its own DNA. However, the retrovirus cannot be controlled fully and can sometimes lead to unpredictable outcomes. The low efficiency in creating the induced pluripotent stem cells show that much more basic research is necessary to understand the many rare events that lead to the making of the induced pluripotent stem cells. One of the main advantages to induced pluripotent stem cells is that they come from adult cells. Compared to the embryonic stem cells that are taken from young embryos that are cultured in vitro, Removing this ethical concern could potentially allow the researchers to receive more federal funding and support, compared to the induced pluripotent stem cells that are taken from an abundant source of somatic cells that are located in an adult human. James Byrne, a faculty member at UCLA involved in the development of induced pluripotent stem cells, said that this also takes out the chance of being inflicted with graft versus host disease. This disease happens when a patient receives stem cells from a donor other than themselves. The stem cells themselves contain T cells, which help to protect the body by recognizing foreign or out of the ordinary things in the body and attacking it. For example, the T cells attack cancer cells, which is how stem cell treatment works to treat cancer. However, the donor cells may also attack the patient's healthy tissue and organs, which can cause the organs and tissues to fail altogether. Being that there is an abundance of somatic cells in the adult body, we can use the patient's own somatic cells and completely negate the possibility of the graft versus host disease. There are two main downsides in the use of induced pluripotent stem cells. The first being the reproducibility and production of the induced pluripotent stem cells. I have another quote from Charles Goldweiss saying, however, the original methods of reprogramming are inefficient, yielding iPSCs in less than 1% of the starting adult cells. So the main problem is that we can't make the induced pluripotent stem cells at all. We only have a 1% chance in yielding them. The second problem of induced pluripotent stem cells is that the retrovirus that researchers have used to integrate the stem cells into the cells of the patient. The problem is that when in certain situations, the retrovirus can have unpredictable outcomes. Sometimes the retrovirus has been found to cause cancer in studies experimenting with mice. These two disadvantages show how much more research is necessary for induced pluripotent stem cells to become able to be used clinically. We have come a long way from the first discovery of all types of stem cells. However, induced pluripotent stem cells are the future. With more basic research of the rare events that come to create them, and research pertaining to the retrovirus to either find another strategy to reprogram cells in the absence of a retrovirus. However, to do this, we as citizens must look to lower regulations and increase funding to increase their findings. The goal is to soon have induced pluripotent stem cells being used clinically. Once this is done, scientists will then be able to generate patient-specific stem cells to either treat or cure disease or an ailment that they might have. Thank you.